Hello, everyone, and welcome to today's webinar, Expi293, New Tools for Structural Biology and Difficult-to-Express Proteins. Presented by Dr. Jon Zmuda, Director of Cell Biology, Life Sciences Solutions Group, Thermo Fisher Scientific. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. This presentation is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. This presentation you are about to see was previously recorded, but I would like to remind you that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Dr. Zmuda will be answering questions via the contact information you provided at the time of registration. If you have any trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I would now like to introduce today's presenter, Dr. Jon Zmuda. Dr. Zmuda is a Director of Cell Biology at Thermo Fisher Scientific in Frederick, Maryland. Jon leads a team dedicated to discovering and developing new technologies and products useful for cell biology applications, including transient protein expression, advanced cell culture, and rare cell analysis. He received his PhD in cell biology from the University of Maryland, College Park. Dr. Zmuda will now begin his presentation.
Today I'd like to talk about some of the new tools that we've developed at Thermo Fisher, focusing a little bit on Expi293, but because insect cells seem to be having a bit of a resurgence at this conference, I'm not afraid to put in a few slides on our XBSF chemically defined insect system as well, and some of the new application data that we've generated within that system. So just a quick reminder, about 7 or 8 years ago when I joined Thermo, our vision really was to provide these first generation high expressing transient systems across the three most typical cell lines for protein expression, Expi293, Expi2, and XBSF as about a year and a half ago. And of course, the Expi293 has been a workhorse system for many labs for many years. This is obviously based on a high expressing 293F cell variant. And typically we can see antibody yields up to about 1 gram per liter in Expi293 in about 5 to 6 days, with a lot of antibodies expressing the 100 to hundreds of mg per liter range. But really, again, what you see is this broad panel of proteins expressed in the 293 cells. So it's really, the system's great for antibody screening, reagent or immunogen generation, for structural biology applications, membrane and intracellular proteins, and really, as of the last decade or so, moving into AAV and lentivirus production as well as the predominant host for those viruses for cell and gene therapy. Expi2 has been an absolute workhorse for antibodies. Anything with an FC, an FAB, fusions, bispecifics, multispecific antibodies has been a choice of many researchers. And then the XBSF system on the insect side of things, often a great alternative to either of the two mammalian systems. So again, part of our paradigm was that we'd be able to get high levels of expression of any protein, regardless of whether it expressed well in one or all of the systems. And so for the insect system, there's certainly, there is a play for intracellular proteins that are toxic to mammalian cells, as well as vaccines, VLPs, and also for large-scale AAV production. Insect cells, SF9s, are quite popular today as well. So what I'd like to do is just introduce you to some of the new tools that we've developed for Expi293. Over the last year, early 2019, we launched a number of these reagents, particularly focused for structural biology, but certainly having overlapping functionality for other protein expression tasks as well. And so what we'll talk a little bit about today, will introduce the fact that we do have CGMP banked Expi293 cells that are available for licensing for both protein or virus production. And then within our Expi293 structural biology module, we've launched 3 new cell lines, so all based on the high-expressing Expi293 parental line, the GNT1 negative cell line that Kenny spoke of earlier today, and Expi293 inducible cell line for modulating your protein expression, as well as a combination inducible GNTI negative line that's popular for many membrane protein researchers, as well as lastly, a methionine-deficient transfection and labeling kit. So really our first foray into doing metabolic labeling within these high-expressing mammalian protein expression systems. So just to go through the Expi293 GNT1 negative cell line, as mentioned previously, this is a knockout of the GNT1 enzyme that prevents the cells from going from this high mannose or MAN5 glycoform to the more complex forms of glycans. And what we see here is that this cell line, this GNTI knockout, retains essentially all of the expression potential of the parental Expi293 cell line. And when we compare that to the ATCC GNT1 knockout cell lines that have been suspension adapted into freestyle media, which is quite common in the literature, we see about a 30-fold improvement in protein titers compared to the ATCC cells, which also tend to be fairly clumpy and a little bit less easy to handle, whereas all of the Expi293 derived cell lines that I'll talk to you today have very similar growth characteristics as the parental line. And what you see on the bottom left is just looking at the glycan profiles for native IgG expressed in the Expi293 system, where we have a pretty typical mixture of the G0F and G1F, G2F glycans. When you take a look at the same material expressed in the GNT1 knockouts, we have essentially exclusive MAN5 glycoforms as expected. And one really beautiful representation of this is in a GPCR that was expressed CB2, cannabinoid receptor 2, where you can see 4 different conditions expressed in the native Expi293 cells, and to the right of that in the GNT1 knockouts, where you get this very beautiful homogeneous band of glycan-regulated protein. And this actually has 2 different functions. One is certainly, as Ken called out earlier today, for structural biology applications, it's ideal to either have homogeneous small glycans or the ability to very easily release those and completely release those with PNGSF, for example. And secondarily, this work done with a collaborator at the NIH also indicated that purification of these proteins of this particular GPCR was much easier when we had this homogeneous glycosylation pattern. So these cell lines do a really great job at controlling that glycosylation, but also keeping very high expression levels. Then jumping over to our Expi293 inducible cell line. So this is a cell line made with sort of the tried and true TET repressor technology, where the cell line has been stably generated to express very high levels of the TET repressor protein. So when you have the Expi293 inducible line, you simply then need to mate it to one of our TET-on plasmids, so pcDNA 4 or 5 TO vectors, and then that will allow you to have control of your protein expression with the addition of TET. And you can see that on the upper right, in fact, using a GFP-fused CB2 GPCR membrane protein, that you can modulate the expression level of the protein quite nicely through the addition of different concentrations of TET. So similar in a manner which in the past had used filler DNA to cut the amount of plasma DNA and thus reduce expression levels. That can also be done with a single step in terms of the titration of your tetracycline. On the bottom, you can see representative images. So we have very low background from the inducible cells when they're in the absence of TET. You can see maybe a couple cells in here that are fluorescing green, but very high expression levels, and potentially even with some subcloning, this could come down even further. But again, looking at a comparison to the existing T-Rex 293 TET-inducible cell line, we've got almost a 10-fold improvement compared to that cell line. And again, when we fully induce these cells, we're as good or even slightly better than the parental Expi293 cells. So a lot of potential. There's a lot of different reasons that one may want to use an inducible cell line. Certainly, I'll show some case studies on membrane proteins where it is proven to be able to significantly improve the expression levels of some of these membrane proteins. And again, just looking at another GAG-M cherry expression, again, very tightly regulated in the absence of induction and very strong fluorescence in the parental or the fully induced cells. And then the combination of these cell lines, so based off of the GNT1 negative line, we then made that a double mutant so that we have inducibility, again, control of a glycan modulated cell line. So again, essentially a single peak at man 5 where we expect it to be. And again, comparing the dual mutant, the inducible GNTI negative cell compared to T-Rex, we've got about an 8-fold improvement in terms of inducibility. And these cells actually, the background here is even a little bit more tightly controlled than the single mutant. Again, showing very nice modulation of signal, GFP, CB2 in this case, with the dose of tetracycline added. And then lastly, from the new reagents is the methionine-deficient Expi293 labeling system. So this is, of course, it's just a single amino acid deficient, so there is still some limitations in the applicability of a single amino acid, but it is a first step in terms of being able to do these high titer expressions with a metabolic label. We've generated data either using a 13C methionine variant or selenomethionine, both very common in the structural biology world for NMR, for example. For the 13C methionine, the cells really don't know that they're in it. They grow perfectly fine. You can employ any number of strategies to label your cells by either passaging them continually in the 13C, which could get a little bit expensive, or simply we've also done time zero where you just spin the cells down, wash them, or replace the media, and you can still get very high 90-95% incorporation ratio of your 13C methionine. Selenomethionine, a little bit different story. It's well known that that's fairly toxic to cells. So we do take a significant hit on titer, but if this is compared to some of the legacy systems where those titers would be close to zero, 200 mg per liter is still quite good. So again, taking a significant hit, but still being able to have a fair amount of protein there. In the case of selenomethionine, the cells cannot be maintained or cultured in that, so we employ typically a spin down. We'll starve the cells in the methionine deficient media and then simply add back the selenomethionine at the time of transfection so that the cells can incorporate that into their protein. And shown below is just a mass spec representation of the native peptide at 917.9 and with the selenomethionine, the molecular weight shift of that particular peptide. So I'm just going to go into a couple case studies. Membrane proteins are obviously a hot topic out there. GPCRs, there's about close to 900 of them, and about 30 to 40% of drugs that are approved today target a GPCR in one form or another. Only a few of them have had their structure solved, and so there's a lot of interest in expressing GPCRs. But there are certainly drawbacks to doing so in bacterial systems that have traditionally been employed, especially the lack of the post-translational modifications that through some of our collaborations and hopeful publications in 2020 will show the importance of having mammalian post-translational modifications on certain properties of these GPCR proteins that may be going overlooked when we're expressing them in the prokaryotic systems. But I just wanted to start with one case study. This is a GPCR AA2AR. It's an adenosine receptor. And I'm going to show data for expression of this in the Expi293 parental cells, the GNT1 negative cells, as well as Expi2. Actually, I'm not going to focus too much on Expi2 in these presentations. As I'll show you in a couple slides, the 293 cells seem to do a great job of expressing membrane proteins. The CHO cells seem to really like expressing antibodies. They don't do as well with the membrane proteins. But for each membrane protein, there's a lot of different considerations you need to take when you're setting up and determining optimal expression conditions. And in fact, I'm only showing a subset of them here. But in this case, we have AA2AR directly fused to GFP and a his tag on the C terminus. And we find that this generally does a good job of helping to establish optimal conditions for the expression of your GPCRs, with one exception is that you have to watch out for the optimal harvest time, because a GFP will tend to sort of diminish in fluorescence past about 48 hours in the cells, whereas if you then look to express and purify the non-GFP version of the protein, you probably have a little bit of a wider window. So for example, 48 to 72 hours is often the sweet spot for most of these proteins. If you look at the GFP-related fluorescence by 72 hours, it starts to come down significantly. But aside from that, it's been a fairly good marker for optimizing membrane protein expression. And typically the GPCRs that we've expressed with it linked to it have retained their activity as well. So just in this small DOE, here we're showing, looking at different doses of DNA and different concentrations of our enhancers 1 and 2. Enhancer 2 for Expi2 would be the Expi2 feed, but we looked at modulating all of these different conditions to identify the optimal parameters for this particular GPCR. And without going into all the bars here, what you're seeing on the x-axis is the plasma DNA concentration from 0.25 to half a microgram to 1 microgram per mil, and then with no enhancer 1, with half of the recommended volume, with 100%, and with 150%. So these conditions here actually represent the Expi293 catalog conditions, so they work quite well for both GPCRs as well as your typical secreted or intracellular proteins. And here we've assessed the expression levels by flow cytometry after 48 hours. And what we've seen with the with the Expi293 GNT1- cells is that we had best expression when we were using relatively higher or normal levels of DNA at about a microgram per mil, as well as the presence of the enhancer 1 to boost expression. And again, these are quite consistent with what we have in our catalog recommendations. Now, with the y-axis being the same, this is Expi2 for this particular GPCR. So very much about 4-fold reduced expression of this GPCR, and it's independent of how long we let the Expi2 cultures go out. So these are representative of the highest expression level that we saw in the Expi2 cells compared to the Expi293 cells. So again, I think this is it continues to confirm our thought process and recommendations around Expi293 as being a very good cell line for expression of membrane proteins. So if we look at some of these optimized conditions, so we went through the Expi293 GNT1 where we showed that using higher or a more standard amount of DNA, so 1 mg per mil, coupled with 100% of the enhancer from the expression system gives us the highest expression levels. And in contrast, however, for the parental Expi293 cells, we see it's actually a bit flip-flopped in that the parental cells can't handle as much expression of this GPCR. So we actually lower the DNA amount to get the highest titers from the Expi293 cells. As we go up to a microgram per mil, we actually lose expression of the GPCR. And again, these GNTI negative and inducible cells are clonal derivatives of Expi293, and we do know that there is variety in the clones that come out, the subclones that come out of the Expi293 population. And then again, Expi2 with the same y-axis here. And what we found in Expi2 is that it actually is beneficial to just go ahead and express the proteins without any of the feed. It's really not necessary to go for 48 hours, and that will give you the best conditions. And then I mentioned some of the work we've done with the inducible cell line. So what our strategy for the toxic proteins or some of these membrane proteins, for example, where you really want to bulk up your biomass to get as much protein as you can. So we looked at transfecting the Expi293 cells, and then adding enhancer 1 and 2 as expected, and then harvesting at 48 or 72 hours. And then we did for the inducible line, we transfected, and we added the TET as well as the feed-in enhancer at the typical 22-hour time point. Or in green, we held off 1 extra day, and we allowed the cells to bulk up in terms of their density. and then we added the enhancers and the induction at 48 hours. And what you can see is, again, firstly, looking at this drop from 48 to 72 hours, I think that's what I had mentioned earlier, tends to be an artifact of the GFP more than the expression of the protein. But what we found is that in multiple cases, and from the last slide, what I was showing here, we got almost a fourfold improvement in the amount of membrane protein expressed. And for two other GPCRs, we had about a 1.5 or 1.6x fold improvement by employing this later induction strategy. And what you can see is that when we run the standard protocol, these cells at the time of addition of the enhancers and induction, we were at about 6 million, so the cells roughly double from 3 million to 6 million. In the case of the ones where we held off and induced later, we were almost double, right around double the cell density. So certainly some of this improvement in expression is based simply on biomass, but in terms of membrane proteins that are expressing in very low concentrations, the more biomass you can have in a small volume, the more cost-effective your reactions are. And just to give you an example for some of these GPCRs, CB2 that we've done work for, we've typically had what are considered to be very good titers in the structural biology community, somewhere in the 3 to 4 mg per liter purified range for some of these GPCRs. So getting actually quite good expression levels with some of these clones in the system. And so now I'll go into one other case study very similarly. This is for optimized expression of ion channels. This is the KCC2 ion channel. And again, this goes into a little bit more detail. This is work that we did with collaborators at Calixar in Lyon, France. And here you see it's a much more detailed optimization where we've looked at two different vectors, so a CMV and an EF1 alpha promoter, different DNA amounts, different cell lines, different temperatures, and the purification tags on either the N or C terminus of the protein. And you can see, quite dramatic differences in these gels depending on all of these conditions. And where we landed, again, we did not see great expression out of the XP-CHO cells, but our best conditions ended up being the Expi293 system with we use the pcDNA5 TO vector just to give us flexibility to potentially couple that with the inducible cell lines in later slides. And in the case of this receptor, shifting down to 32C was most beneficial. So again, each one of these membrane proteins really needs to be looked at in its own and conditions optimized for it. But these are some nice EM data from purified KCC2 that exists both as a dimer and a monomer. And you can see some of these monomers and dimers in the EM graphs, and you can see that on the SEC as well. But what we ended up with was very nice aggregate-free KCC2 that was able to be concentrated well for downstream structural biology studies. While XP-CHO expressed much lower, the quality of the protein was still quite good. And we also saw similar results for a sodium channel as well that we tested side by side. Another example of the GNTI negative cells, so you can see the difference in how they run on these gels. Again, in a number of these bands, the high and low glycosylation indicative of the GNT1 negative cell line. Again, protein quality was very good for that cell line, as well as for the TET-inducible cells, where you can see no TET and then titration of TET induction and KCC2 signal going up. And lastly, these are just some early data of work that we've been doing in our lab to boost expression levels of our membrane proteins. And this was just some internal development work that we have ongoing right now, where we were able to increase the expression levels 4 to 5-fold of KCC2 compared to the parental Expi293 system, which for the structural biology world is actually quite a feat, because again, every fold that you have of protein improvement in these very low expressing proteins means the systems become more cost effective for the researchers. So we'll take just a few minutes to go through a couple slides on the XPSF expression system. So this is an SF9 based insect system. It utilizes the first chemically defined insect media for high titer production of proteins in insect cells. And like all of our Expi systems, it's built along the premise of combining different reagents together, optimizing them in a systematic approach so that we get synergistic benefit of protein yield from the systems. And I think very similar, as Sabine called out earlier today, one of the other hallmarks of this system is we have a transfection reagent to do highly efficient suspension transfection of our backmid, such that we make P0 virus. We can make large quantities of P0 virus that's very good for protein expression, and you can go right into an expression run. The other thing that's quite interesting as well is that while you're making your P0 virus, it's such an efficient amplification of virus, and you're also making your protein of interest, so you actually have an opportunity to look at your protein of interest from this same flask that you're making your P0 virus to get some indication on whether it's going to express well or not in the insect cells. So again, taking this from 12 to 20 days, down to 6 to 10 days, and I think in the insect systems, because of the upfront labor in making the viruses, any days that can be chopped off of that workflow are very valuable. So just some quick performance data for proteins and cell growth. So this is the Expi-SF9 cells. These cells are very high density. They will grow to 20 million cells per mil in a shake flask, so almost as high as the CHO cells. And you can see just a representation of the comparison to all of the different a panel of different insect media that are all yeastolate containing. So we've got very, very good growth of the cells compared to them. And just 3 proteins that we show here compared to all of the existing panel of yeastolate containing media versus the Expi-SF system for these 3 proteins. 3, 4, 5-fold improvements. Please, visit our poster later to get more details on that. And again, GPCR expression in insect cells also quite common. In this case, we had about a 10-fold improvement in the Expi-SF, the high-density system, compared to a traditional SF900-2 workflow. So we were able to get very good expression of membrane proteins. And what's new with the Expi-SF system? So for large scale, as I mentioned previously, for vaccines, VLPs, AAV, baculovirus is of significant interest to manufacturers. So we do have an AGT version, so a advanced granulation technology version of the media for scale up. We talked a little bit earlier today about scalability of the insect systems. We've generated some data. A 10-liter run in a 22-liter wave bag compared to shake flasks, and these cells and the system itself work quite well. So we've got very similar growth kinetics in the shake flask versus the wave bag. We tend to see that the wave bag usually lags about a day behind in terms of titer and some of the kinetics compared to the shake flask. But for 2 proteins you can see here, we've actually got quite similar and respectable loss of titer, minimal loss of titer in the wave bag compared to the shake flasks. And then lastly, some data of the Expi-SF system adapted into a 2-liter stir tank bioreactor for expressing AAV. Again, we see very similar trends between the shake flasks in red, the bioreactors in blue for VCD, viability, cell size. We get very nice expression of our AAV and the representative proteins here, looking at a standard low-density 900-2 or 3 protocol compared to the Expi-SF system. Very significant improvements in AAV titers, and our AAV is infective on susceptible cells, in this case recombinant AAV2, and again, much more information at our poster. And lastly, I'll wrap up just in terms of some examples of enveloped and non-enveloped VLPs in the Expi-SF system. Again, compared to our traditional system, very significant fold improvements for these proteins. But I will certainly say, as was mentioned earlier today, the insect expression world is much more like of the Wild West. So everybody has their best cells, their best media, their best virus, and it depends on which cells you make which cells you use to make your virus in which media. The combinations are really daunting, and I feel for the speaker earlier today, because it is very difficult to really get a good competitive assessment on different insect systems because of all the variables. It's much more much trickier than with the transients and your relatively well-defined plasmid DNA. Lastly, I'll wrap up. We've got 3 posters here today, 45, 46, 47, one focusing on Expi293, one focusing on the membrane proteins, what we talked about today, as well as the XBSF system. And with that, I'd just like to wrap up and thank a broad panel of people, all of the R&D team that's not in Canada, that's based in Maryland, as well as the folks that have helped the purification, the analytical, Henry and Natasha, who are here today. Please, if you have a chance, I'm sure many of you know both of them. Our field application scientists, Matt McKenna is here as well, provides excellent scientific support across the East Coast, and our colleagues at Calixar and Lyon for the KCC2 and NAV 1.6 collaboration studies. And with that, I think I'm right on time, Carlos, so I will wrap up and thank you very much for your attention today.
A big thank you goes out to Dr. Jon Zmuda for his excellent presentation. Now, if you do have any questions you would like to ask Dr. Zmuda, please type them in the Ask a Question box located on the far left of your screen. Dr. Zmuda will be answering questions via the email address you provided at the time of registration. I would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. That's all for now, and we hope to see you again soon. Have a great day.